Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. M. Lois Tiffany Endowment, and by gifts from friends of the program.
Today I'd like to tell you a little bit about a new area in which physicists have recently become active, and that's nanomedicine. Let me start out perhaps with telling you a little bit about what nanomedicine is and why do we really need it. In medicine generally we'd like to cure diseases. And to do that we generally have roughly two options. We can either do it with drugs or we can do it through surgery. And if you look at the length scales here, they are very disparate, the two. Drugs usually are small molecules, most of them a nanometer in size. Some larger ones we can get up to the size of proteins here. For instance, the insulin, hexamer, gets up to 10 nanometers in size. But generally, drugs are not any bigger than this. On the other side here, surgery. Until recently, we were mostly limited by the skill of the surgeon, how steady is his hand or her hand. And well, with recent advances, we can now do microsurgery with micromanipulators, so eye surgery or so down to perhaps 100 microns, perhaps a bit better than that. But as you can immediately tell, there are four orders of magnitude in between here, the 10 nanometers and the 100 microns, in which we have very little ways for medical intervention today. And this is a gap that one would like to fill. On the diagnostic side, it looks a little bit better. We'd like to find out what is it that makes us sick. And again, we can look at the molecular level here. For instance, if you measure blood sugar, you measure the concentration of a very small molecule. A sugar molecule is a very small molecule. If you try to go a little bit larger, we can look at proteins, for instance. Again, a Western blot is a typical case of a molecular assay, molecular diagnostics, here in this case for HIV testing. What we can do here is essentially look at proteins that you have in your blood serum, for instance, and tell you whether you have these kind of viruses. So this is something on the molecular side we can do, again, up to about 10 nanometers. On the large scale here, what we have, of course, medical imaging, X-ray, MRI or so. We can do this down to, again, 100 microns perhaps resolution if you're really good. Then we can go even further down. We can take a biopsy and we can look under a light microscope with it. And in the best case, perhaps with a light microscope, 1 micron resolution or so is what we can get there. So here, a typical histologic sample here has here a scale bar of 20 microns. So again, here we have at least two orders of magnitude in between the molecular diagnostic and anything we can do routinely with a regular microscope. And so we have this nano gap here, in particular on the therapeutic side, but also on the diagnostic side. Now, there can be two reasons for having such a gap. One could be that we don't know yet really how to work on these intermediate length scales, and the other one could be that there's just nothing interesting there, that there's no reason to bother. And so let me show you, there is plenty going on the nanoscale. So here, many of you have probably seen this here, a typical eukaryotic cell. It has plenty of subcellular structure. So anything, here we have the nucleus, and here a close-up of the nucleus. Here you have in pink the DNA wrapped around histone cores. Then it's transcribed here, some transcriptionally active DNA, and so on. So there are lots of things going on the scale below the size of a cell and above the size here of the individual protein molecules. So certainly of interest to look there. And is it medically relevant? And here I just pick an example that is studied in our medical school here quite a bit. One of these nanostructures here that they're looking at quite a bit are so-called immotile cilia. So little protrusions from cells that can do a lot of functions, mostly sensing. They are sensors for mostly chemical sensors, mechanical sensors, and they have a typical size here of 200 nanometers. So they are right in our nano gap here. And if they malfunction, they have been implicated in a lot of diseases here, many of them clinically very relevant. Cystic kidney disease, for instance, we have blindness, liver fibrosis, and a lot of other things. So clearly, we have nanostructures that we need to look at and study and find out what we can do to hopefully address some of those diseases here. Now, we have to see how can we bridge this nano gap here. And we have there a couple of options, or if you ask different people, we get very different answers what we can do on this to bridge the nano gap. If you could show us the video. This is Hollywood's version of it. Essentially, Hollywood is suggesting we miniaturize a submarine and just go into the body and look at these cellular and subcellular structures. Unfortunately, they don't really quite tell us how to do it. As physicists, usually we take a somewhat different approach. Generally, before we set out to do anything like this, we'd like to understand the physical laws that actually govern the systems that we are looking at here. So very much like Newton, we had to understand gravity first before we could fly to the moon. So similarly here, we have to understand the physical laws that govern nanoscale biological systems first before we can even reasonably think about addressing some of the medical questions. So that's at least where we come in from a physics point of view. And talking about Newton, governing laws here. Newton's laws, you're all familiar with those. We have something that drops, for instance, force equals mass times acceleration. The sphere just keeps accelerating, we have a constant force. This is because inertia dominates. We also have a different regime where friction dominates. If I drop the same sphere here into this viscous glycerol solution here, then what happens is the sphere is no longer accelerating, but it drops at a constant velocity. So what's happening here is we have a different law of motion here. It's no longer force equals mass times acceleration, but force equals friction times velocity. So we have two very different equations of motion here. And now we have to think about what do we do, what is relevant, what is applicable in biological systems on the nanoscale. And we can make a very simple argument what should happen if we go to smaller and smaller and smaller systems. We can think about, in this case here, mass is a relevant parameter, and it scales as radius or size of our structures cubed. So if we cut the size in half, the mass is cut in 1/8. So it goes down very rapidly as systems get smaller. For friction, it's different. Friction scales linearly with radius. So if the system size is cut in half, friction is cut in half. So what we see here is mass reduces much, much more rapidly than friction. And therefore, if we go to nanoscale systems, we usually end up with systems that are completely dominated by friction and not by inertia. So this is really the equation of motion that governs all these nanoscale biological systems. Mass is usually irrelevant on that scale. And, well, that has a few consequences. One is if you look at oscillations. For instance, if you want to keep time, you usually build a clock, and most mechanical clocks just oscillate like this one here, or your grandfather clock, you have a pendulum swinging, And that's what we usually do in mechanical clocks to keep track of time. But if now, for instance, a cell wants to keep track of time, and you were to do the same thing here in a viscous medium where we have the other equation of motion, so where friction dominates and not inertia, what happens now if I drop it in here? Oops. I guess. Anyway, I have another one. This is a bit heavier. It doesn't work quite as well. But, well, yep. We tried it a couple of times. This hasn't happened before, but let me see if I can. Okay, let me see if I can get this one here fixed. Okay, so outside it still bounces nicely. And if I now drop it in here and let it bounce, hopefully without, it does not bounce anymore. So it stops dead in its track. That's what we call an overdamped harmonic oscillator. And if it's not oscillating anymore, we have a hard time using it as a clock to keep track of time. So just shrinking down a grandfather clock to cellular dimensions wouldn't work. So we have to find something else. And well, the something else can come from chemistry. Let me show you that one, how that looks like. Let me first put the goggles on, just to be on the safe side here. So what you'll be seeing here are chemical oscillations. The flask is changing its color a few times until at some point we run out of reagents. Now it's turning, or should be turning yellow again, and turning blue again, and keeps oscillating for a while. So chemical systems can oscillate, and therefore we can use them to keep track of time, for instance. However, this is fairly inefficient. Having the entire cell, for instance, oscillate chemically would use a lot of reagents, a lot of energy. So usually biological oscillators are much, much more complex. And in fact, complex mechano-biochemical systems. And let me show you an example of the simplest one that I'm aware of that they built up in Alex Ninfa's lab in biological chemistry here at Michigan and put it in E. coli just to show what a minimum clock could look like. And I don't want to go into the details of how this one works, but essentially what we need here is several genes here, 1, 2, 3, and they are all regulated by a fairly complex promoter structure here that can turn them on or turn them off. And essentially what they are doing is here, one produces a protein, codes for protein that activates this type of promoters here. The other one generates a protein that inhibits them, and they mutually activate and inhibit each other, and we end up with oscillations here in our product protein that we are looking at. So this is an example here of a minimal clock that Alex Ninfa has designed and put in E. coli. And what you see here is generally this very complex promoter structure. That's DNA where proteins can bind. And let me tell you a little bit about those, because that's now where really very interesting physics is happening. Most of them here have some very simplified versions, actually depend on bending the DNA and causing either transient contact between two proteins or long-living permanent contact here, such protein-mediated DNA loops to form. So one of these proteins, for instance, binds to two sides on the DNA molecules, bends it into a loop like this, and then, for instance, it starts to generate one of the genes, to express one of the genes, generate the protein. For instance, a protein here on the other side that would permanently bind to the DNA molecule, bend it in a loop, and so on. And with this type of structures, these are here very much simplified structures, but they can become quite a bit more complex. We can have multiple loops in one that control one single gene, and with those, actually, you can code logical functions. You can do AND, NAND, OR, and so on. So this way, with this type of loop DNA structures, cells can almost do computing, or in the other case, building these clocks. And so these type of protein-mediated DNA loops are extremely common in biology. And so to understand this, first thing we need to do here is to look a little bit into the physics behind bending and stretching DNA. And that's something that my group is very active in. We are trying to understand the physics behind deforming DNA, forming such loops, and so on. And first thing you need to realize here is what's really governing the mechanical properties of DNA. And this is what's called entropic forces. And so what I'm going to tell you is, when we are talking about, for instance, stiffness of DNA, I'm not talking about we are twisting or pulling on molecular bonds here. All of this is statistical physics. And so we can introduce this just with a very simple example from classical thermodynamics that probably many of you are aware of. If you just have gas molecules, put gas molecules randomly into volume, into a room, chances are that they are more or less evenly distributed. Very unlikely, if you put them in randomly, that they all end up, let's say, on one side of the volume, one side of the room. It's just not going to happen if you just put them randomly there. This is something that's highly, highly unlikely. This is something that's very likely, so usually you end up with something like this. And in fact, if you want to force the system into a conformation where you have all gas molecules on one side of the volume, like this one here, you actually have to apply a force. You have to physically push the piston in against a force here to get them all onto one side here. And this is here an example of such an entropic force. This is an unlikely conformation compared to this one. And to go from here to here, we have to apply an external force. We have to supply energy to the system here to push it into this conformation. And very similar things hold for polymer molecules, like DNA, RNA, proteins to some extent. Again, what we are doing is we have now usually we have these chemical bonds here between two adjacent monomers. And the bonds themselves, they are very, very hard to stretch. But what they can usually do is they can rotate fairly easily, like this. And so if we just leave it completely free up to itself, what's going to happen is the polymer will form a random walk. So one monomer pointing this way, next chemical bond is oriented in some random direction here, going in a different direction, and so on. So that's why polymers usually, on their own, tend to coil up. This here is something relatively unlikely because in this case, we would say all bonds have to point more or less in the same direction to get to a stretched conformation. So the random coil is highly likely. The stretched conformation is rather unlikely. So if this here is my DNA molecule or my polymer molecule in general, we have many conformations, many different ways of aligning the bond angles to have a random coil like this one here. But ultimately, we have only one possibility, all bond angles lined up in a line to really stretch out your DNA molecule. And as I said earlier, a force is required to force a system from a likely conformation here to an unlikely conformation. So when we talk about stretching DNA, we are not pulling on bond angles here, trying to on the bonds themselves, trying to elongate them or anything. All we are doing is we are just reorienting them and force them all to be oriented the same way, like in this case, and that takes work to do it. We recently did a little demo experiment to show how a random walk usually leads to a coiled conformation like this one here. So we just made a human polymer here right in front of Randall. So we had a couple of people just lined up, holding hands, all of them blindfolded. They should be walking around now completely random, but they can't let go. And what happens if they do that? Oops, what happened there? I want to show the movie. Okay, so this is now we start here with this stretched conformation, and now we just let it go. So everyone is just moving around randomly, bumping into each other, and equilibrium, in this case, equilibrium is a coiled conformation like this one here. Equilibrium is not the line we started out with. Okay, so that's what's behind stretching DNA, and bending DNA works actually very similarly. But now let me actually show you a little experiment. This one was historically the first one how people have stretched DNA molecules and actually measured how much force it takes to stretch a single molecule out. Essentially what you can do is you attach DNA molecule, a relatively long DNA molecule, on one side to a surface, a cover slip, usually microscope cover glass, and on the other side to a small superparamagnetic particle. And then you can just use a magnet, pull on it, or even use another magnet to twist it, and see if you can watch under a microscope how this here, how you stretch your DNA molecule. And Gerhard here has actually prepared a sample. This is always a little bit of a black magic, so it doesn't always work as well as it should. Really dealing with these DNA molecules in microspheres is a bit tricky. And what he's now going to show you is actually, can we switch to the view? Okay, so what we have here now is microspheres, and many of them are attached to the cover slip now via a relatively long DNA molecule. So they can move around locally a little bit by Brownian motion, but they are tethered with a DNA molecule like this. So they move around, but are attached on one end. And if we now bring a strong magnet near it, we should be able to stretch them out a little bit and align them. And now if we let go again, it assumes their random conformation. They can move around again. And if you can let go again, yeah. So that's how the first measurements were done to measure the elasticity of DNA. Just attach one end to a cover slip, magnetic particle on the other end, and stretch it. Thanks, Gerhard. Okay. No. Now, we can do this a little bit fancier today, get better data, and we usually use optical techniques. Don't want to go into all the details of it, but the idea is still more or less the same. In this case, we have two microspheres with a DNA molecule in between. We use lasers to move the microspheres around, and then we can do fairly accurate measurements of the force extension curve of a DNA molecule. Initially, it's more or less linear here, and then we start stretching. We approach its nominal contour length. At some point, you cannot stretch it anymore, and the force goes up dramatically. Perhaps worth mentioning, the force scale here is piconewton, about a piconewton or so. In this case, we used DNA from a bacteriophage, so a virus that affects bacteria, and its contour length is about 16 microns out here. And we can compare to a theory that has only statistical mechanics in it, just probabilities of that the bond angles line up and nothing else. So nothing about how stiff a bond is or anything. And this is here the red curve and experimental data, and here the theory fit perfectly. So this is really a case where we see purely entropic forces, can measure them and understand them. So that's in the, so this is a kind of experiments that we are doing on a fairly regular basis in my lab. Let me show you another fun one. This one here, it's not playing. Here we have again a DNA molecule between two microspheres, an even longer one. And here we are trying to tie a knot into a DNA molecule. So stretching it out as far as we can and come back relatively quickly and hopefully see if we can go through the loop. It's almost like playing a video game. You have a joystick and move it through, and if you're reasonably skilled with it, you actually manage to make a knot here. And, you know, so we can stretch it and relax it a bit here. Let's see, it should get a little bit brighter. Yeah, so here still the knot is still there. We can again try to pull it tight. So this is the kind of games we can play with modern micro and nano manipulation techniques. Now, back to these complicated regulatory structures where protein bind to DNA. Well, we start out with a very, very simple one here. We'd like to keep it as simple as possible. So we just look here at a system where a protein binds to two specific sites on a DNA molecule, like this one here. And we'd like to actually see these loops forming and breaking down. And once we can do that, then we can change various parameters. We can study, for instance, effect of the size of the loop on loop formation rates and so on to really understand the underlying physics of these regulatory structures here. The way how we do this technically, it's very similar to what you just saw there. We attach a DNA molecule and a microsphere to a cover slip. The main difference here is we use fluorescent microspheres and an evanescent wave to excite the fluorescent microspheres. This way we can get not only XY in-plane position, but we can also get axial position because when the microsphere moves close to the surface, the evanescent wave here is brighter, so we have more fluorescence. If it's far away, we have less fluorescence. So we have not only XY position here, but we also have the brightness of our particle to get the Z dimension. And so we can get all three dimensions. And let me show you this here. This is then how it really looks under the microscope. So we have here one that moves quite a bit around. This has a fairly long DNA tether attached, more like this here. Here, this one here has either a short tether or the protein is bound. And so we can use the motion of the microsphere here as a measure for the length of the DNA molecule in between, or the effective length here. And for instance, then here's an example. We can actually see, if we look at the amplitude of the motion here in a solution where we have the protein in there, the protein binds to the DNA molecule. That means the tether all of a sudden, so initially it starts out like this here, moving freely. We have a large amplitude of the motion. Then the protein binds, forces this here into a small loop, and now it can move much less here. Just move around a little bit, and that's what we see here. Protein has bound, protein falls off again, the motion recovers. Also axial dimension here. Initially we are sticking out quite a bit here, then all of a sudden we move fairly close to the surface here, and then out again. So we can look at this type of protein DNA binding events, how these structures here, typical size 100 nanometers or so, how they form, break down. We can play with the parameters, loop size. We can change the sequence of the DNA in between. For instance, engineer kinks into the DNA and really see how does that affect loop formation. We can, for instance, with the magnetic beads that you saw earlier there, we can apply mechanical tension to the DNA and find out, for instance, does it matter if the DNA is under tension? Does that affect these regulatory structures? So one of the questions that we're asking there is essentially, what's the role of mechanics in regulating biological functions? So that's much of what our lab is doing. Let me now shift gears a little bit and tell you one other thing that's relevant on the nanoscale, and that's to look at transport on the nanoscale. What we need to do is in a cell, you have seen this complicated structure earlier on, molecules have to get to their destination wherever they are needed, so they have to move around in a cell. And well, macroscopically what you do when you do chemistry, like what we did earlier over here, we just stirred things, or we shake a test tube. So that's what you do conventionally if you want to get, in chemistry, a molecule to its reaction partner or so. Well, there's a little problem with that on the nanoscale. And that has again to do with that on the nanoscale, we are operating in a regime under extremely low Reynolds numbers. So what I mean with that is essentially, and I'll show this in an experiment in a second, is that you cannot, on a small scale, you do not have turbulence. Usually when you shake something, you induce turbulence. Little vortices everywhere. That's why things mix efficiently if you shake something. On a nanoscale, we don't have that. There's something, some of you probably have heard about it, in fluid dynamics called the Reynolds number. It's a dimensionless parameter that essentially tells you whether you can have turbulence or whether all flow is laminar. And it depends on the size of the system, the velocities, densities, and viscosities. And let me show you a little bit about this, perhaps in this experiment here. What we've set up here is a capillary, and we create a little dye jet here, some dye solution coming out here into the fish tank. And if we let that go, what we see is, oops, can we go with the camera? Perhaps you can align the camera. So right after the nozzle here, the dye jet is coming out completely straight. There's not much happening here. So it's right here, it's completely what's called laminar flow. And if we go further out here, what you see, if the dimensions here get larger, so we're getting further away from the nozzle, then all of a sudden we're beginning to see turbulence out here, all these little vortices. And that's exactly the effect that I just mentioned. On short length scales, all flow is laminar. You need large length scales that turbulence can develop. And that means mixing on a cellular scale is extremely hard. And if you only have this laminar flow, if you don't have this large scale turbulence out here. Let me show you what I mean with that mixing is hard in a laminar flow regime. If we can, can we? Yeah, let's perhaps do this one first and then do the other one. So what I have here is two concentric plexiglass cylinders with a very viscous solution in there, just to make sure that we are again in this low Reynolds number regime where friction dominates everything and where we cannot really get turbulence. And what I'm going to do is I inject a line here of a fairly strongly colored dye here, and then I'm going to shear it. So I induce a little bit of a flow in this cylinder here. Let's see. Mm-hmm. No. Let me see if I can do this here. No, not working yet. Yeah, no, just. Maybe. Having any dye in here, or is it just bubbles? Okay, there's the dye. Okay, you can see the line here. And now I'm inducing some flow, and the only flow that we can have in this type of low Reynolds number situation, laminar flow, just by spinning one of the two cylinders here. So I'm trying to distort my line here. And it's still a line, so I have a very, very hard time even mixing this here, but it's already spreading out a little bit. Now, how well have we mixed? Usually, if you think of something as mixing, you would think it's irreversible. It's impossible, for instance, if you mix two solutions that mix well together, to separate them again. So how about this one? So if I reverse the rotation here, and so I just spin it backwards, so the entire thing here is very reversible, and my original line has reappeared. So that's part of what I mean with mixing is very, very difficult on the molecular and cellular scale here because all flow is laminar. So we are very often in a situation like this one here. Well, we are on small scales, so maybe we have another thing that we can do, and we can hope that diffusion will just take care of it. Well, diffusion, you just put essentially your molecules in and just hope that Brownian motion takes care of it and distributes them slowly and evenly everywhere. There are two problems with it. One is it's very slow, even on a cellular scale with very small molecules. I prepared here a little gel that has about the consistency of a typical biological tissue, if you can get the other one, and put yesterday a little dye drop on it. Can we get a little bit? Can we get a better contrast? Or can we get a better contrast of this? Yeah. So essentially, I prepared a gel here that has about the consistency of typical biological tissues. Put a little dye drop on it yesterday, a very, very small drop. And by today, it has spread to perhaps a centimeter or so. You see, well, I hope we could get a somewhat better contrast, but essentially what you see here is that we have this little dye drop here in a day has just spread to a relatively small area. So even for a small molecule here, diffusion is still very slow. It's also inefficient in another way. If you think of how many different molecules you have in a cell, 30,000 different types or so, generally you don't want to spread them out completely evenly over the cell. You would like to have them really where they need to go and only where they need to go, and not completely everywhere. What you get in a case like this here, if you let this here sit, let's say, for a couple of weeks or even longer, maybe it spreads out all the way. Probably not the most efficient way of running an organism. So nature has come up with something else, and that's active transport. It has created little molecular motors, little nanomachines essentially, that can bring cargo to wherever it's needed. So here again, this immotile cilia has on the inside little microtubules and then proteins, kinesin molecules, and then a cargo can dock to this kinesin molecule, and the kinesin molecule can actually walk along the microtubule and bring the cargo wherever it's needed. So here's an artist's rendering of this. This here is the kinesin molecule. It has two head groups that can actually walk on the microtubule. I'll show you this in a second. And then on the other end, it can carry any kind of cargo, small vesicles with molecules in them or so, and bring them to wherever they are needed. So that's essentially nature's way out of the problem with the laminar flow and the inefficiency of diffusion. Nature built nanomachines here really to do the job and bring things into the right place. Let me show you two little movies here. This one here is a very schematic one, if I can, how this here works. So here is a little sketch of. So here you see what's happening is it binds first. Here we see the binding, and now once it's bound, it uses a fuel here, ATP, hydrolyzes ATP to ADP, and then makes a step in this type of hand-over-hand motion that you see here. So next step, and so on. So this is how this little nanomachine actually moves along a microtubule, and it does that actually quite efficiently. So it's a wonderful little machine. And well, of course, this is something that certainly fascinates the engineers, a machine like this. So in the moment, there are really people trying to build little machines based on those molecules. So here's a very simple example in the moment. This here is not much yet of a machine really with it, but I can still show you what this is. These are these microtubules here, stained, and they now went the other way. They put these motor molecules on the cover slip. You can see them, they are equally distributed everywhere here. And what happens now is the microtubules actually move on the motor molecules, just the other way than what you just saw in the first movie. And they tend to glide along here. So here you see them, the microtubules moving on the immobilized motor molecules and crawling around. And people are actually now using this to make, for instance, little racetracks or so. Can you make a little rotary pump by letting this type of molecules move around in a little circular racetrack or so? And so that's actually work that's also going on here in our biomedical engineering department, that type of things. So here you may be able to learn a little bit from nature. Now that we are beginning to understand physics and engineering on the nanoscale, can we actually do something with it? Ultimately, we'd like to create drugs for medical intervention to actually cure diseases. And this is something that's still very much in the infancy. Let me talk for a moment about an effort here at Michigan, what's coming out of our Nanotechnology Institute for Medicine and Biological Science over at the medical school. So they say, okay, we need to make bigger structures in a controlled way that we can actually essentially make almost little nanomachines here. And we know quite a bit about polymers. I showed you earlier how we can study and manipulate them on a single molecule level. So what they are doing is they synthesize a very special kind of polymer, dendritic polymers. So they branch in a very controlled fashion, and more branches, and then you get these dendrimers here in the end. And now what they do with it is now put everything on it that you would like to have in a controlled way. So for instance, groups that recognize folate receptors that are overexpressed on cancer cells, so they would be stuck on these polymer scaffolding. Then you can put drugs on it that would actually kill those cells. You can functionalize them in a way, for instance, that you know that you just killed one, that it, for instance, releases a fluorophore or something. So you can do a lot of things here on these nanostructures that have been synthesized in a controlled way. And they make even more complicated structures now. So here's an example of two dendrimers linked together with a DNA molecule in between. So you can make things that look even closer to real nanomachines, smart, multifunctional therapeutics. That's what we hope to get out of it. We aren't quite there yet, but I think we are getting close. And well, on this hopeful note, actually, I'm ready to conclude here. So I hope I've been able to tell you that there are important biological and medical problems on the nanometer scale. And we have this big nano gap. We have these almost typically 3 orders of magnitude between conventional medicine and molecular medicine. And in between is where we need to go. We are in the process of understanding the relevant physical laws, what's going on this scale. So we need to rethink how we think about equations of motion. So instead of conventional Newton's laws, we have this now friction-dominated regime here. Inertia and mass don't matter anymore. we have to think in new ways about entropy, new ways about mechanics on the nanoscale in general. And we also have to develop experimental tools to really study physics on that scale. So you saw the magnetic tweezers. I had examples of the optical tweezers. We need to continue that. And ultimately, in the end, we hope that we can this way develop novel therapeutic approaches that can actually cure diseases. But this is still very, very early on, and we are not quite there yet where we can actually use this in clinical practice. And well, with that, I'd like to thank the Physics Demo Lab staff for setting up all the demonstrations. In particular, Emily has worked very hard over the last week to put all of this together, and thanks a lot and for indulging all my wishes to change things in the last minute. And my research group, in particular Gerhard here, who showed the magnetic tweezers, and also the NIH Institute for General Medical Sciences. They not only provide my funding for much of my work, but even in the times of a very constrained federal budget and increased congressional pressure to really rush things into clinical practice to come up with cures tomorrow, they are still very supportive of this very physical, very fundamental work because the hope for payoff is so great. So a big thanks here to NIH that they are still believing in this. And thank you for coming.
Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. Lois Tiffany Endowment, and by gifts from friends of the program.